Okay, we'll go ahead and start it. Okay, hi everyone, welcome to another Tropilunch. Today we have Joshua Benjamin. He's a doctoral candidate in the Department of Biology, pursuing a PhD in zoology at the University of Florida. He holds a joint master degree in limnology and wetland management from Boku University in Austria and in Netherlands. Additionally, he is the founder and manager director of the Freshwater Guardians NGO in Kenya. During the 2023 and 2024 academic year, he served as the president of the Tropical Conservation and Development Program at the University of Florida. And recently, he was awarded the prestigious WWF Russell E. Training Fellowship from the Education for Nature program. Joshua has received other competitive research grants, including the funding from the Ruford Foundation and the Explorers Club. Currently, Joshua is a committee member in the International Union for Conservation of Nature, a Species Survival Commission, contributing both globally and within Kenya. He is an avid freshwater ecologist, traveler, naturalist, and amateur wildlife photographer. So thank you, Joshua, for your presentation today, and the floor is yours.
Thank you so much for this wonderful opportunity for me to present. It was the last minute call, but yeah, I think I got this. So let's talk about this small cryptic species we call the aquatic insects, or if you like, the macroinvertebrates, and how we could use them, or how we are trying to have this kind of a model from one of the river cases in Kenya and trying to upscale it to more like a national level where we could adapt the use of these insects for biomonitoring in Kenya and the East Africa. And so, as we all know, for those who are from the ecological background, is this trend of the species being more accumulated across the equator. That's where we have much of the concentration of many of the species in this region. We call it the tropics, which is basically the region between where the equator crosses a little bit up and south, and that's more of the Amazon region in Brazil, all through Kenya as a country as well, where the equator passes. And that region is usually high in species or biodiversity. But also, so we're trying to advance the slides. Okay, and so the tropics are really rich in species, but compared to these two ecosystems, both the terrestrial or the freshwater, we see that there's a trend that in the freshwater, these ecosystems are mostly kind of threatened in these tropics because they're experiencing more of these kind of threats, and mostly issues of climate change, pollution, and all that. And so this graph shows you that there's a possibility to lose more species in water than those which live on land. And so from the species which you've seen that are high in the tropics, a lot of threats also prominent in the tropics. So this recent paper which also confirms that we are almost losing a quarter of our global freshwater species, and probably mostly in these regions of the tropics. And also the organisms which I study, which are the macroinvertebrates, the dragonflies, are also facing this kind of a threat of extinction. And further is that we also seeing that the climate change projections and threats could be more of a concern along the same place where we have more of the biodiversity happening along the equator or the tropics. And so that is more of the background, trying to build a case as to why I do what I do. And we do this in this system in Kenya, which is a transboundary river between Kenya and Tanzania. And this is the famous river where the wildebeest migration every single year happens. And these animals, those are our kind of bisons in Africa, which are the wildebeest, are crossing from Tanzanian side all through coming into the Kenyan side. And they have to cross this river again back to that region. And so it's one of the river which is perennial river system, meaning it's the only natural source of water for Serengeti Masai Mara ecosystem. And so it's really important for the wildlife. The people living in this region, over 1 million people, are mostly dependent on this river, and many more. And so it acts like a model of what we want to study. It's one of the huge rivers with mega fauna in Africa or in Kenya with lots of people numbers, more than 4,000 numbers of peoples. It's a river with lots of these reptiles, the crocodiles, and it's really just an interesting river to study. And so if you have been to Serengeti or Masai Mara in Kenya, you probably mostly came there for that, which is more the charismatic terrestrial big animals. You came there, you enjoyed seeing these elephant, leopard, and whatnot. But I'm sure you might have crossed this river, which is the Mara River, and which you probably didn't pay much attention to it. But it's really important in this ecosystem, and we are trying to tell the story of this river, which is understudied in the form of what form of biodiversity they exist and what role the river might be playing in this kind of ecosystem. And so I'm gonna quickly introduce you to this stream theory, which basically is the river continuum concept. And it's pretty quick and easy. I'm gonna make it so simple that when the river is flowing from upstream all through to the mouth of that river, the river is characterized by a lot of things happening. The riparian vegetation, the organisms might also be characterized by the width of the river and how fast it's flowing and things like that. But typically when the river is at the source where it's forested forest, the light is unable to penetrate through, right? And so the river cannot do the primary productivity. So it depends for its energy source from externally from the river, which is the shading of leaves into this river, which forms like the energy source for the river. And that's include a lot of allogenous materials coming in from external source, and that forms like the energy source for the river. But as it moves downstream, it becomes bigger in length. It opens up, the canopy open up, the light can penetrate through, and now the other subsequent processes such as primary productivity would happen, and that's going to be the food source for these algae, diatoms, the insects will come in, the fish will come in, and all that. And so we are trying to test, this has mostly been tested a lot in the temperate region, which is the regions where we experience the 4 seasons here in the US, in Europe, but mostly not in the tropics. And also, the rivers in the tropics probably do not conform to that kind of a theory because we at most have a lot of these different stressors happening. And we are working in the Mara River where there's a lot of land use issues, there's a lot of subsidy input into the river, and we're also having a lot of anthropogenic disturbances in the river. And so we are trying to measure all those and try to see how our organisms assemble and distribute along this river where this kind of theory is not like a case or usually not mostly tested. And so what we have done so far, I think early this year, late last year, we wrote a book chapter about what kind of threats could be in this catchment which we study. And that is the main Mau Forest catchment, which is the main source of the water for this river. So we have written a book chapter. It's somewhere online, and basically here we're trying to describe the many different threats which we're experiencing in this catchment, and which mostly led us to this idea that there are only mostly 3 of those particular threats in the catchment. The ones I talked before, the land use change is like a thing of a concern. Probably over the past 15 years, a lot has happened in the catchment. Issues of the wildlife subsidies through the crossing the wildebeest. And once they die and they are decomposing the river system, they are adding a lot of nitrogen, phosphorus into the system, and that could be a signature for kind of higher nutrient elevation at that particular section, and that could fertilize and, of course, influence photosynthesis in the river. And so this is just a proof of this kind of threats and why we are of concern in this river. That's been the main source of the river. You see it somewhere inside the forest, but there's a patch where probably there's some agriculture or some settlement going on at the source of that river. There's a lot of sedimentation, as you can see. The water is always murkier throughout the year, meaning there's a lot of agricultural activities probably happening upstream. and so everything is washed down, and hence the color of the water. There's also a lot of different sources of pollution, but the most documented one is this issue of potential for mercury contamination in Tanzanian section of this river, because the river flows from Kenya all the way to Tanzania to Lake Victoria, the second biggest lake. And so there's a huge artisanal and industrial gold mining happening somewhere in Tanzania. And what is happening is that they use mercury as an element for amalgamation for getting the mercury. But later on, this waste somehow comes in these massive big pipes. There were three of them. Can't tell why the other one is missing, but somehow they are discharging whatever they are calling the treated waste back to this river. And we're also trying to test or try to answer questions about the possibility of ecotoxicology in the river. How much of different heavy metals do we have existing in the river? And that's a different other chapter I'm working on. But we are trying to measure the different levels of trace element and heavy metals in water, in sediment, in fish, and macroinvertebrates, and just trying to understand that kind of a pathway, how much of these trace elements really have concern and what component. So maybe that's a talk for another different time. But for today, let's focus much on the insects. And so also the effect of the wildlife subsidy input, where after these animals cross the river, as you can see, there's a documentation of a lot of them, they are dying in the system. Those are vultures feeding on those carcasses, but what is happening is that they could stay there for a long time, and the bones are persistent in the river and in the ecosystem, and they could have this kind of legacy effects where they continuously continue releasing phosphorus or nitrogen to the ecosystem. And so I'm gonna take you to our system where we do this, and this is the catchment area of this river from the source all the way to the river flowing to Lake Victoria. Pay attention to these pink, purple, dots, purple. So the four of them, it's basically what we are trying to do. So I'm working with a data set where we are trying to compare data over intra-annual variation. So we have data sets spanning out for 15 years, and we want to understand within these 15 years what has been happening in the catchment in terms of land cover change, in terms of water quality, in terms of the species composition, in terms of what drivers are the most important to influence the flow of that. And most importantly, as what has happened within this 15-year period of time, has it impacted these communities, which could be important to be used as indicators for environmental health of the rivers. And also the last bit will be trying to show you the kind of work we are doing in Kenya. Since we already have the results, we already know what is happening in terms of rivers, but how can we interpret this data set to mostly kind of trying to reach the communities and help conserve the rivers in Kenya. But these four dots are the same sites where we have sampled multiple times within a span of 15 years. Make sense? And so I talked about the crossing of the wildebeest and all whatnot, but this area is mostly where they are doing this. So this is their path. They come through this from Serengeti to Kenyan side, and this is potentially the sites where they might be crossing this river again back to this side. And this might be the area where we might have the effect of those kind of subsidy legacies happening in the river, those sites there. So for those who are interested to see what the land cover of this area could be looking like, it's mostly, this forest has been shrinking over time, as I'm gonna be showing you some evidence of what the forest cover has been changing over time. But it's interesting that this is the only remaining part of the forest for this important river system between these two countries. And as you move down, there's a lot of agriculture happening, the build-up areas and all that. If you're interested with what the rainfall for this ecoregion might look like, of course, the watershed area is where we're experiencing much of the rainfall in the catchment compared to the other parts of this watershed. So this is basically what I explained, but 15 years ago, 2000 and 2008, Amanda Spalaski, who is my advisor during her PhD time, she worked in the same river system, and they collected data spanning up to 2 years during 2008, 2009, and that's the data set we are trying to compare with what I'm doing currently when I came to do my PhD from 2021 to 2023. So we're sampling the same sites, using the same equipment, standardizing everything, and we want to compare that data set and try to understand if these organisms, which are indicators for can be used for monitoring, if they have changed. Are we losing any species? Are they coming in? Is there kind of turnover being happening on these sites? What is happening really? So that's the kind of question we're trying. And these are the organisms we are talking about. Maybe if you have never seen them, but if you have been to a river, I'm sure at some point you might have been tempted to pick a stone or a rock. When you turn it, you're sure you're gonna see some crawling little uh, organisms, and those are the guys we're talking about. Only that these images have magnified those organisms. They look so big, but they should be small and probably be seen by the aid of a microscope sometimes, but you can also see them with your naked eye. And so what we are seeing in this catchment over time, as you compare these two maps spanning that period of 15 years, what has happened in the catchment is that we have lost a little bit of forest. This is the table. We don't probably have time, but this is the mean. This is the most interesting table, which might be of interest to you. What we are seeing, there's been increase in agriculture. So 15 years ago, agriculture was at 56, but now that number is short. It's going up. In terms of forest, it used to be at that percentage, but now it's dropping. So as the agriculture is increasing, the forest is going down, and that tells you probably they're encroaching on the forest, or probably they are cutting some trees somewhere for them to enhance the agriculture activities and all that. But that was the most exciting, that agriculture is still going up as forest is going down. And what does that then mean to our system? The water quality also, we have seen there's been a spike in nitrate is coming up. Turbidity is coming up. Have you seen our water is more turbid than it used to be? The total dissolved solids have gone high in this catchment. Conductivity is way up now, and things like that. So there's a lot of that happening already in the catchment, and we are seeing the water is kind of deteriorating, moving from good to a little bit losing its quality. And so what is happening to the organisms which indicate the water health? So 15 years ago, it's this small sample you see this side, and now it's this big one. How you interpret this kind of a plot is that 15 years ago, these sites, the four of them, the ones I showed you with the purple dots, almost had the same composition of species. And so those sites almost were similar. That's why they are clustering together. But now we have moved far away from each other, which means the same sites are having kind of species turnover. There's something being added or removed from what it used to be, and that's why they're different from each other. So that's like the take home, that over the 15 years ago, there's been a change over the taxonomic composition of these macroinvertebrates over that period. If you're taxonomists interested to see what organisms changed, there they are, but I won't dwell much on that slide. In terms of diversity, we have also seen there's some variation, because diversity is used as a form to show how healthy the ecosystem is based on what organism you're studying. So the diversity has been varied, but most interesting currently we are seeing is like we are having an increase in diversity. How is that true? We are losing land cover, the water is deteriorating, but again, the diversity seems to be picking up. What does that mean? In terms of these organisms which indicate the water health, especially these three categories, the mayflies, the stoneflies, and caddisflies, 15 years ago, these organisms were more abundant in the upstream sites, which that makes a lot of sense because we perceive the water to be a little bit better there. But currently, the same organisms are lower in the upstream sites. It's like these organisms we are trying to lose them from the sites where the water used to be pristine. So these organisms which are good indicators for water health. And so the same thing with the percentages of that number, because the first graph was about the abundance, the numbers, but this is now the percentage of those numbers of those organisms which indicate water quality. And 15 years ago, the sites upstream, which is pretty what should be, was higher, but now we are seeing that we are kind of losing those sensitive taxa. And so if you might be interested to know what is attributing those kind of changes, we have this variable or environmental variable which might be important for you. That in the catchment, elevation is pretty important in classifying how the organisms are distributed, the flow of the water, the land covers, and these other different variables. But what does that mean? I've shown you really good results of what is happening in terms of insects, but how can we translate that to more interdisciplinary kind of approach? And that is what I'm going to be talking in this other phase, which mostly relates to what you do here at the Center for Latin America a lot. And so using the same organisms globally in the US, Europe, and Africa, these organisms have been used over time. There are people whose job is just dedicated to analyze the water quality or try to assess or monitor rivers across the globe just by using these organisms alone. And so going back to the case like where I come from, the Nairobi, if you have been to Nairobi or Kenya, you have possibly have heard of the Nairobi River, which is this river. It's actually named after Maasai name, which means a place of cool waters. That's what it used to be. Like when they established Nairobi some years ago and they wanted to like get like the maximum benefits of that river to the city, that it could be the river source which is providing them with really clean water with all what they needed during like let's say 60 years ago. But right now this is the situation of the river. The government of Kenya is trying to use a lot of money and try to rehabilitate the river at this point. And so as a complementary, what I do in terms of studying these insects would come in handy in as part of this process, that we can use those organisms which I study to try monitor as the river gains its good health, transitioning from the bad water quality and now trying to move to the good water quality using the organisms I study. And so the reason why these organisms might be used is because of these reasons. They don't move a lot. They are mostly just stationary, and so they can accumulate a lot of toxicants in the system if they have been there for, let's say, 1 to 3 years, and every single time there's been discharge, they can have that signature to tell you what their bodies comprise of if they've been able to uptake a lot of toxicants. They are pretty easy to sample. They have rapid life cycle, larval stages, pupal adult stages, which is pretty okay to use them. They indicate water quality and the long lifespan, which is between 1 to 3 years, and which could help you get a snapshot of what has been happening in the catchment for 3 years instead of just doing the water quality. Because once you do the water quality, it's just a snapshot of what has happened probably last night. Did it rain or not? Did we have flood? And so what you're measuring in water is just a signature of a short period of time using water quality. So the insects come in handy because they could tell you like the long-term story of what has been happening over time, right? And so there's a lot of this theory or is evolution of these different methodologies or protocols on how to use the insects themselves to monitor the streams. The US, they've been using that. I mentioned about that. The UK are also leading in using these insects as indicators. But on the side of Africa, we have South Africa, which are the leaders of using this now. And theirs is as this it's coined SAS, which is an acronym for South African Scoring System, which is basically a protocol where you have all the names of the organisms in there, right? And then we have a column which has the QV, which is basically the quality value for every particular species or organism. This value ranges between 1 and 15. If you have a higher value, it means you really pretty much love to be in clean water. You are sensitive to pollution, but if you have a lower number, 1 to 3, then you love to be in pollutant tolerant system. You could persevere being in dirty water. And those are what these values are. So every organism has been assigned a value. And so using this protocol, every time we go sampling and calculating these quick matrices, which is basically the SAS score, which is a combination of just these numbers of the organisms who you will have found at that site, divided by how many numbers of different species you found at that particular site, then we are able to come up with this kind of a plot where you will have the average score taxa, which I explained, you get it by combining all the quality values of that particular site, divided by the average number of how many species you got. And this side is, of course, those number of the SAS score we just talked about. So this helps you to calculate these two particular matrices, which can be plotted here. What does this then mean? So your values, because you've already calculated them, you somehow plot them. You're gonna see how much was your SAS score. Maybe it was somewhere 120. And at this particular site, what was your, let's say your SPT? It could be something like 5. So where 120 meets your 5, within that plot, that's where your site lies within, and that's what the class is gonna be. And what does that then mean? So if you did all that and numbers are aligned within those numbers, this is what the interpretation means. So if you align here, it means your site is natural, it's not so much disturbed, there's nothing much coming in terms of that. But as you decline in quality, then you are losing a lot. In case there's pollution coming in and all that, you start now losing that number to, just like in school, when you're scoring an A, it's great. But when you are ending up scoring a C, a D, and F, things are not great. And so what we've been doing in Kenya, quick, I just want to show you the comparison of my data 15 years ago and now using this kind of a protocol, what the results were, in order to show you why it's applicable. So the same 2 dots, 15 years ago, all the sites scored an A, which meant like those sites were like pristine or natural. But of course, not pristine, but they scored really high numbers of an A. Right now, the same sites 15 years ago have dropped in quality, and they haven't, from an A, majority of them are scoring to a B. So in terms of management, if there's no intervention done in this river, it could continue losing its ecological balance, and it could end up scoring a C, a D, whatnot. So packaging all this information together to make meaningful impact in Kenya, we already do a lot of these macroinvertebrates data, but sampling and all that. But how is it helpful to the communities? So we have initiated some outreach programs in Kenya where we want to impact change. And so we are assured, we have done research and we have seen these insects are changing. And so we want to improve the health of the rivers in Kenya. Nairobi River is just one example of many rivers in Kenya which might need a little bit of rehabilitation. But if the government of Kenya is now using approximately around 40 billion to rehabilitate one river, how about things become worse? Climate change is an issue, and in 50 years from now, every river needs to be rehabilitated. How much money do you think the government is going to spend? There won't even be money for doing any form of development, not just in Kenya, but in any country, right? And so we want to be part of the change and try to prevent it from going that way. And so we've started approaching the next generation of leaders, the young people in the society who could really be a change. And so we target the university students because the techniques can be easily understood by this kind of a group and by everybody. It's easier to access them because I don't need to pay anything to go to those schools. I just need to pitch the idea to these universities, and then they invite me to talk to the students who are taking environmental courses or any conservation related courses. And so we usually have series of workshops where we're going to introduce them to the idea of monitoring. We take them to the river at some point, show them all how it works, like how do you do it the proper way. We bring samples back to the lab, we help them identify them, and by that, we are believing we are changing the perspective of these young people because they're going to get interested with what we do. They're going to have interest in freshwater, probably, and they might even persuade graduate programs in this kind of a field. And so during my practicum through TCD, I was able to do some of these activities in Kenya at 4 universities, and it was received well. I've trained more than 200 university students. And so just not about the university, but we have extended this now to like target group again in high school, because those are the guys who are coming up now to university level, and we want to get them excited even before they get to university, right? And it's also some programs we are doing in high school level. Talk about the elementary or primary school level. We have also now started programs to have them also even more excited at that level and have them like talk about these insects. Probably not everybody has seen them, and they really get so excited looking through the microscopes, and they're like, "Oh wow, I didn't know in rivers we have such kind of insects which live there. And so they have been received well. And so we have also extended this to a group of people who might not be in any of these institutional programs, like in high schools or elementary or universities. Do we still reach them? Yes. Through citizen science, a lot of rivers in Kenya, they have this kind of association where they have the local people of the communities being part of that kind of water resource users association, and they are the people who are championing the conservation of that particular river. And so what we do with them also, they don't need to be in any educational institution, but we also work with them. We do the same things, workshop over a period of days, just making sure they are doing the right thing, understanding the protocol, and easing it for their understanding. And just not in Kenya, but we have now extended this mostly to East Africa countries. We were invited this year to go to Tanzania. We have done this somewhere in Tanzania. And not just in Tanzania alone, but also recently we did this in Uganda. And it's more like an East Africa kind of an approach on how we could really use these kind of methodologies to improve our rivers there. And if you've talked to me, you must be seeing that I'm really passionate about freshwater systems, wherever they are. Mm-hmm. And so that drove me to trying to be also a change at an individual level as Joshua from Kenya, and what can I do to my country with the knowledge that I have. And then I came up, I'm a founder and the managing director of this NGO, which I've founded now in Kenya. We are up and running and actively involved with activities happening there. But the main goal is still to try to project the activities we've been doing in high school, wherever now, as an institution which has this kind of a mandate for a long time period, you know? So what we just started is not something which we think we need to finish that, but with this organization, we can always now go back, train them whenever they want, and try and be like the change in Kenya and all that. So yeah, look up onto it. You can scan the QR code. We're still looking for collaborators, potential partners, and all that. And with that, I want to acknowledge everybody who has been part of this process, my advisor, my lab, my everybody, the permitting processes and the different funding organizations for sure. Thank you so much for your time.